In the previous lessons, the components of the innate and adaptive immune system has been discussed in detail, as well as its significant physiology that is necessary for survival. We have learned that the immune system with its components are the actual foot soldiers of the body against the invasion of a variety of foreign materials such as bacteria, viruses, parasites, allergens, and many other types of immunogens. For this discussion, I will be making a determined efforts to deal with the other components of the innate immune system. The complement system incorporating its major pathways, the different molecules that regulates the complement activity as well as the deficiency of its components and disease association, and of course, the different assays that determines complement abnormalities in the immunology, serology, clinical laboratory. The complement system. The complement is a composite of innate immune espionage-like system that plays a central role in the defense body mechanism against foreign materials. It is discovered by Dowsett. Consists of a series of serum proteins which are heat labile in nature, that are normally present in the body and whose overall function is mediation of inflammation. Collectively, these proteins are mostly enzymes of or proteinases that are a major fractions of beta-1 and beta-2 globulins. Complement proteins makes collaboration to form a cascade that performs salient effector functions, which are the following: opsonization by C3b, of anaphylactoxin by C5a, C3a, and C4a, chemotaxin by C5a. These all lead to the lysis of cells by the membrane attack complex. Nomenclature of complement components. The components of the complement system are numbered C1 to C9. The lowercase Arabic letters after the number is designated for protein that is smaller in size as a product of cleavage of a larger precursor by proteases. Studies show that these plasma complement proteins are being synthesized in the liver. Pathways of the complement system. The complement pathway can be activated in three various ways, depending on their context. These are classical pathways, alternative pathway, lectin pathways, and each pathway leads to a common terminal pathway. According to studies, an immunocompetent individual, the alternative pathway is permanently active at low levels to survey for the presence of pathogens. This is due to spontaneous generation of bioinactive molecule C3 to activate C3H2O or the water. Now let's go on to a more detailed discussion of each of the mentioned complement pathways. First, we have the classical pathway. Classical pathway is the main antibody-directed mechanism that triggers activation of the complement, meaning to say an antibody will attach to an antigen to form antibody-antigen complex in order to activate the pathway. In other words, an antibody-antigen complex is a requirement for classical pathway activation. Usually, immunoglobulin G and immunoglobulin M are the required antibodies for activation. For additional information, the classical pathway is the first activation cascade to be described among the three. The principal components of the classical pathways are C1 through C9. The classical pathway has three major stages: the recognition, amplification of proteolytic complementation cascade, and the membrane attack complex. In the recognition stage, the recognition unit is the C1 complex, which is composed of three subunits: 6 molecules of C1q, 2 molecules of C1r, and 2 molecules of C1s. The first step is the initiation of the pathway triggered by the recognition by complement factor C1 of antigen-antibody complexes on the cell surface. When C1 complex interacts with aggregates of immunoglobulin G with the cell surface, two C1 associated proteases C1r and C1s are activated. Take note that a single immunoglobulin M molecule is potentially able to fix C1. However, Immunoglobulin G requires two molecules for this purpose. The C1s is a serine protease and can activate the C4 and C2 complexes. The C4 and C2 complexes will be cleaved by C1s, producing C2A and C2B. C4A and C4B fragments, respectively. The resultant C2A fragment joins with C4B to form the new C4B2A enzyme, or the C3 convertase of the classical pathway. The C2B and C4A fragments from the C2 and C4 component are lost to the surrounding environment. In amplification or activation of the complement, the classical pathway C3 convertase will activate the C3 molecules. Take note that the C3 molecules is the most abundant complement component, and it depicts the most salient step in the process of the complement activation. Upon activation, the C3 will be cleaved in two fragments, C3A and C3B. The C3A will not combine with C3 convertase, but it will attach to the surface of the mast cells and act as anphylactoxin. This will stimulate the immune cells to release chemicals such as histamine that will eventually attract other immune cells and as a result, increases the rate of protection. The other bound C3b molecules not involved in the C4b2A3b complex will bind covalently to the cell membrane of the pathogenic cell and will act as opsonin, which carries the process called opsonization and renders it the cell susceptible to immune adherence. Now the other C3b will combine with the C3 convertase, forming the final proteolytic complex of the complement cascade known as C4b2A3b. Membrane attack complex. To initiate the formation of the membrane attack complex, the C3b will attach to the allosteric site of C5, which is the first membrane attack unit. C5 will be cleaved, and as a result, the C5a and C5b fragments will be produced. The fixation of C5b will call upon the formation of C7, C6, and C8. When fully assembled in the correct proportions, C7, C6, C5b, and C8 will form the membrane attack complex. The C5b-C6 complex is hydrophilic in nature. However, with the addition of the C7, it has additional detergent and phospholipid binding proteins as well. Once the membrane-bound C5b, C6, C7 is relatively stable and can interact with C8 and C9, the C5b, C6, C7, C8 complex will be polymerizes C9 to form a tubule pore, which spans the membrane of the cell being attacked, allowing ions to flow freely between the cellular interior and exterior. By complexing with C9, the osmotic cytolytic reaction is accelerated. This tubule is a hollow cylinder with one end inserted into the bilayer and the other projecting from the membrane. A structure of this form can be assumed to disturb the lipid bilayer sufficiently allow the free exchange of ions and water molecules across the membrane. Ions flow out, but large molecules stay in, causing water to flood into the cell. The consequence is that the influx or sodium ions and water leads to the disruption of the osmotic balance, which produces cell lysis. Now let's proceed to the other pathway of the complement pathway, alternative pathway. For the alternative, it is similar to some portions of the classical pathway as it also generates a C3 convertase. The major difference of the alternative and the classical pathway is that the alternative is not triggered by antigen-antibody complex, but instead it is being activated by polysaccharide and lipopolysaccharide, example of which are insulin, zymosan, bacterial polysaccharides, and endotoxins. The alternative pathways was first described by Pillemer with his subordinates in the early 1950s. It was previously called a proprotein pathways because I believe that proprotein, a normal protein of the human serum, was the first protein to have a physiological activity in the alternative pathway. The key feature of the alternative pathway is that the first three proteins of the classical pathway C1, C2, and C4 is being overlooked in the cascade sequence. So it starts with conversion of C3 into C3A and C3B. The C3B will bind to factor B, which is a serine protease and active enzyme of C3 and C5 convertase, forming now the C3B B complex. And bound factor B is cleaved by the factor D into BA and BB. So factor P binds to C3B, BB, and will stabilize it. C3 convertase cleaves C3 into a C3A and C3B. C3B joins C3B, BB to form C3B, BB, 3B complex. C5 is cleaved to produce C5B. With the cleavage of C5, the remainder of the complement cascade continues as in the classical pathway. So for the last pathway, we have the mannose binding lectin pathway. Like in alternative pathway, the MBL does not require antibody for activation. Instead, it is activated by bind of MBL to mannose found in bacteria, yeast, viruses, and some parasites. So, mannose binding lectin is a member of a family of calcium-dependent lectins and is homologous to C1q of the classical pathway. And this pathway is very identical to the classical pathway. So in correlation with the serological assays for complement, now let's proceed to the diagnostic evaluation of the complement components. So the proteins can be demonstrated in tissue by appropriate immunopathologic staining procedure as immunologic reactions occurs on tissues. The following assessment for complement components can be used for diagnostic immunology. First one is C1 esterase inhibitor. A deficiency of this protein is characteristic of hereditary angioedema. C1R, C1S, C2, C3, C4, C5, C6, C7, C8. Homozygous deficiencies predispose a patient to autoimmune disease, especially SLE, and to arthritis, chronic glomerulonephritis, infections as well as vasculitis. C1q, this is being evaluated in serum. These decreased levels can be demonstrated in patients with hypoplementemic urtical vasculitis, severe combined immunodeficiency, or X-linked hypogammaglobulinemia. C2, the most common complement deficiency and an autosomal recessive disorder. Half of the patients with high homozygous C2 deficiency have no symptoms. C3, unelevated levels can indicate an acute inflammatory disease. Decreased levels are seen with patients with post-streptococcal glomerulonephritis and in those with inherited C3 complement deficiency. C4, it levels provide the most sensitive indicator of disease activity. C5, associated with increased susceptibility to bacterial infection. C6, decreased quantity of C6 predisposes an individual significant Neisseria infection. C7, decreased level is associated with Raynaud's phenomenon. Severe bacterial infection caused by Neisseria. C8, decreased level is associated with SLE also, and C8 deficiency makes patients highly susceptible to Neisseria infections. So, to conclude the discussion, we know that the complement is not just a simple lytic system, but also a powerful innate immune surveillance tool serving as a sentinel against pathogens, a modulator of the adaptive immune response, and a regulator of host homeostasis. The accumulation of knowledge of this molecular mechanism in recent years has allowed for a better understanding of complement-related diseases. Thank you for watching.